Hi, I'm Sagar Kumar. In this video, I will talk about the Magneto Encephalography sensor, which is very useful for medical science and neuroscience. And now onward, I will call it MEG. Okay, so let's see what it does. So it usually measures the very weak magnetic field generated by the electric current in the brain. So this magnetic field is very small, like in the units of femtosecond. So this is the structure of neuron. So in this structure, there are the dendrites which receive the signals from other neurons, and then these dendrites send the signal to the cell body, and the cell body process this signal and send it to the axons. This axon is the wire-like structure which receive the axon from the cell body, and it decide if the signal will send to the axon terminal this side or not. And it depends on the potential of the signal. If the signal potential of the signal is more than some threshold value, then this axon will send the signal to this axon terminal, and then this axon terminal send that signal to the another uh, neuron. And in this process, the signal will transfer through the ions inside this wire-like structure. And the size of this axon depends on the size of the animal. uh, for different size of animal, the size of this axon is different. Okay, so this is the internal process of the axon, like how the signal pass through the axon from axon to the axon terminal. So in this structure, when some effective potential come to the neuron, so these gates start to work. Like in this structure, there are two regions. One is extracellular region and another one is intracellular region. And these regions are separated by some boundary, and there are three types of gate in this boundary. One is this blue and green and yellow. So this gate allow only sodium to transfer from the extracellular region to intracellular region, and this green kit allow only potassium to transfer from the intracellular region to extracellular region, and this uh, yellow gate allow both potassium and the sodium, but this allow three sodium to transfer to the extracellular region and two potassium to the intracellular region. So let's see how does it work. So when some potential more than this threshold value come to the neuron, the sodium ion start to transfer through these gates from the extracellular region to intracellular region, and the potential gradually increase to this peak. And after this peak, the concentration of sodium in this extracellular region become very low, and in this intracellular region, the concentration of sodium become very high. So then put this, these green gates of potassium is opens, and the gates of sodium closed. And when these gates of potassium opens, the potassium started to transfer from this region to extracellular region, and then the potential start to decrease to the stable value. And then the gate is this gate is gets slowly closed, and then in this process, the potential goes lower than the stable value, which is called the overshoot potential. And to overcome this overshoot potential, this third gate start to open, and this third gate allow 3 sodium and 2 potassium to transfer. Then the 3 sodium from extracellular region goes to the 3 sodium from intracellular region goes to the extracellular region. At the same time, the 2 potassium from the extracellular region come to the intracellular region, and then potassium gradually come to the stable value. And actually, the name of this boundary is cellular boundary, and the movement of the ions through the cellular boundary creates generates the electricity inside the neuron. So in this slide, we will see a video which is the internal working process of the axon. So let's play. So this is the signal is coming from the axon terminal of the another neuron, and this will receive by this dendrites. And the activation potential of the signal is greater than the some threshold. And this is the internal structure of axon. In this structure, this is the gate which allow only sodium to transfer from extracellular region to intracellular region. And when the sodium transfer this voltage increase to the and here you can see the potential reach to its peak, and the gate put is sodium gate closed, and now potassium gate start to open, and the potassium start to go beyond this boundary, and this potential go to the stable value. But due to the delay in the closing of this gate, the potassium overshoot is the lower than the stable value, and now this gate will start. And in this third gate, this allows 3 sodium and 2 potassium, and now this overcome this overshoot. And this process take only few milliseconds, and this is a chain reaction go along the axon, from axon to axon terminal. So as we saw in the last slide, the movement of the ions generates the electric current, and this electric current generates the magnetic field. But the magnetic field which we are talking about is generated by the dendritic current of pyramidal neurons that fire synchronously and in the parallel, and axonal and synaptic current and their magnitudes are cancel out each other. So there are some challenges also in this process. One is the magnitude of this magnetic field generated by the brain is very low, and other is the noise from the outside, like the magnetic field of Earth, which is in the range of 10 raised to power minus 5, and the magnitude of the brain magnetic field is 10 raised to power minus 50. So it will cause some problem. So the solution to the second problem, second problem, the problem for Earth magnetic field is the magnetically shielding rooms, which we call MSR. So there are three components of this MSR. One is ferromagnetic shielding, another one is eddy current shielding, and active shielding. So for the ferromagnetic shielding, we use the ferromagnetic materials like nickel, and this ferromagnetic shielding protect from the low frequency noise like the magnetic field of the Earth. And for the eddy current shielding, we use the thick conducting coils like aluminium, and this protect from the high frequency noise. So let's see how MEG works. So there are three parts in this instruments. One is helium. Helium basically provide the cool environment for the superconductor, and another one is squares, which is basically superconductor, and one is pickup coil, which detect the magnetic field of plane. So let's see what is squares. Squares is the superconductor, mainly it made from some metals like nickel, and when the nickel cool down to extremely low temperature, it becomes the superconductor. And why this is different from some normal superconductor is because of these Josephson junctions. And these Josephson junctions consist of two superconductor separated by some insulating, so that the Josephson effect occurs. And this helium provide the very low temperature so that the metal becomes superconductor at very low temperature. And why we choose helium? Because the helium remains the liquid at very low temperature. And the pickup coil, which detect the magnetic field from the brain, and this consists of two main parts. One is the magnetometer, and another one is the gradiometer. So magnetometers have superior sensitivity to detect the shallow and deep source, but they are more prone to picking up the outside noise. Thus, the magnetometers are most optimal in setting with the limited source of the outside noise. On the other hand, the gradiometer are less sensitive to the which makes them the superior option in location with the number of large-sized electronic devices. and the pickup coils are linked with the quiet Y input coil and are the kept in the superconducting state by helium liquid. The square then produces a small voltage current that can be detected through the through this FFL electronics. FFL electronics is basically the flux lock loop. The electrical output can then be transferred into the digital signal through the optical cables, and this output is usually displayed on the computer systems. Systems connected to the MEG or MEG magneto encephalography. Okay, so thank you. That's all.